We are live from downtown Low Bay. Live. And we're live. We are updated feed. That looks good. That looks like us.
How did you go to the thing last night?
Yes.
You actually not invited?
No, because he said like
You got invited.
little fire. That's great. I didn't know you guys.
Why not be
Oh, we weren't invited.
Oh, you went?
Yeah, because no one was there. Crazy
Oh,
as
yeah,
it
yeah,
is.
sorry. Tony went to cancel.
Isn't the spot like 5 kilometers down there by the rock?
Oh, I guess.
Oh wait, no. 2.5. Yeah, I know what you're talking about. We weren't invited. We were stealing. I'm gonna watch it. Lucas. Pablo said something. Was that nice up there? Yeah. It was straight. We warmed up in the afternoon. Why is that ice? Stomped it for you. That was. Yeah. It was just cheap. So we had to stay on the freeway. But barely on the street. You should come one of these and get it. I felt like I'm close. Not even gonna lie. The last time I read, there's like the lodge, and then you go down a bit, it's like the easy chair there. I did that. I have not watched it. Yeah. It's the one to the Whisky. I think so, yeah. And then once you get off there, you go to that.
Apologies, I can't take attendance because apparently I don't have internet. We'll wait for that to load. So I might as well get started. Okay. Laptops and phones should be away. Your notes should be out. Laptops away. Thank you. I gave you 2 last day. We're going to do 5 of them today. There's more like 8 or 9. But one of them I kind of cheat with. We are today going to look at how DNA goes from being the code all the way to getting the trait. So you have genes. Those genes get transcribed into a form. Then they get translated into proteins, and then they actually make the trait, which might be brown eyes. What were 2 functions of proteins from last day?
Do you have any food?
You just had break, and there was food done.
I had to print out my homework.
2 functions. If you think of protein, what do you think of? Yeah. You can get energy, so that's a new one. It is an energy source. Not a great one. Your body will use it for energy in times of extreme change in your diet, usually for the worse. So for example, if you decide, you know what, it's been the holidays, I've been eating way too much, and I'm going to pick this really extreme diet, I'm going to drop my calories by, 1,000 calories a day. Your body will go, oh my God, we're starving. Who knows when we will see food next? You'll start burning fat and protein. Your muscles will get smaller. You'll actually have a lower metabolism afterwards because it takes energy to keep muscle alive. It doesn't take nearly as much energy to keep fat alive. And so you're lowering your amount of muscle as well so you can survive longer. Your body has no idea that you're doing this on purpose. You can also just, if that happens to be your source of food. So if you are you're the Inuit in the north and you do not have access to fresh vegetables in the wintertime, then you're going to have a primarily hunting meat-based diet. you can just turn it into energy that way. So if you have to. However, if you were not one of the Inuit, you don't know how to do it properly, you can actually starve by eating pure protein. Has anybody ever heard of rabbit starvation? It's not the rabbits that starve. You're a Canadian hunter, you're up north for some sort of hunting expedition, you get snowed in, you make it to an emergency cabin, which dot some areas. There's a little bit of supplies there, but not a lot. And you're stuck there for a while, so you run out of the emergency supplies of food. And while you can't find vegetables in the ground, because everything's covered with snow and they don't really grow in the wintertime, there are a lot of rabbits. So you eat the rabbits. You hunt the rabbits and you eat the rabbits. And you starve to death eating the rabbits. Because you are not normally used to this type of food, you're probably treating it like a barbecue and just skinning the rabbit and eating the muscle. And the rabbits in the wintertime are very lean. They don't have a lot of fat on them, so you're not getting much fat on them. It's just pure protein. And you actually cannot process pure protein for energy unless you get a little bit of a fat with it. So if you're ever in that situation, safety tip, crack open the rabbit's skull and eat the brains as well. It's really high in fat. It'll allow you to use the protein for energy, and then you'll be fine. You'll be fine. You'll have the fat and the protein. You'll survive until you get picked up or the snow melts so you can get back on the trail. You're looking a little disgusted. I hope that doesn't happen to you. But in an emergency, eat the brains. They're probably, if cooked, the safest part. You can also eat the other things like the liver, but if you don't know what you're doing, like stay away from the intestines and stuff unless you know how to cook those. A little gross. Because that's where they keep the poop. So yeah, energy source. That's a new one. What else did we say last night? There's two more. Yeah.
To form
Enzymes. We need to catalyze those reactions. So make reactions go burr. Can I take attendance yet? No. Safari couldn't open the page because the server stopped responding. Eventually it worked. I told you all about neverssl.com.
Yeah.
Yeah. Turns out it's time for me to sign in again, and nothing was giving me the prompt. So, brief interlude. My apologies.
They're just like fake, like hosts.
There we go. Now is everything's connecting, kind of. And attendance. Alexander?
Yep.
Area? There. Ariel?
Yep.
Charlie? No. Zhao?
Yeah.
Guy?
Yeah.
Isaac? Isabel? Jessica?
Yep.
Katie? Kiona? Natsuho? Sihu? Sterling? Jaden?
Yeah.
Tyler?
Yeah.
Confirmed attendance. Nice. Okay, what else do we use proteins for? Yes.
Helps repair and build body tissues.
Yeah, which body tissue?
Muscles.
Muscles. Oh my god.
Oh.
Oh, there it goes. Okay. This is under structure. So it makes up some of the structure of your body. So for that, it will be muscles. Are cells primarily of protein. I also mentioned a couple last day. What other structural proteins are there that make up your body? Sorry. No, fat is not protein. Fat is a different thing than protein.
Calcium.
Calcium is a different thing than protein. That's an ion. You said organs?
Like organelles.
Oh, yes, on the cellular level. I'm talking about body level. So your muscles are made of protein.
Oh, like hair or like nails.
Hair and nails. Your hair and nails are essentially the same thing. Your tendons that hold your muscles to your bone are primarily protein. And then there's a few others like the vitreous fluid of the eye. If you eat an egg, the yolk, a lot of fat. The white part is primarily protein. That's why you can froth it up so much. And if you whip just the egg white, you can do a lot of cool things with it. It provides a lot of structure. The vitreous fluid of your eye is a lot like egg white in its composition. I mean, it's clear, it's a mucousy protein, which is why if you stare into a microwave and it's broken and it's leaking some microwaves, you won't give yourself cancer. That's actually impossible. But you could hard boil your eye in the same way that you can hard boil an egg. It'll turn white and just won't work anymore, and that's relatively permanent. You can't unboil an egg, essentially. Yeah. Again, back to the Inuit. If you are eating non-vegetables and eat vitamin C, also weirdly high in vitamin C in your eye. So don't eat the humans, but raw seal eyes, apparently delicious. I've been told by an actual person, like ice cream. I pointed out, didn't think so, because it doesn't contain that much sugar. And they're like, no, I mean, it's delicious like ice cream. So there you go. I haven't had an opportunity to try it, but apparently a little frozen,
Don't try it.
it's delicious.
I don't like a
Isn't, I'm pretty sure the indigenous hunting practices of the Inuit are definitely allowed.
Oh.
Yeah. There are people who don't like the seal hunt. I think there's regulations on fur and stuff, but really, you want to keep warm. The cute little seal. And you get to remember it forever whenever you pet it. Okay, moving on quickly from that before I say anything else regrettable. We have structure, muscles, hair, tendons. So it makes a lot of the physical structure of your eye. And energy source we're missing two. What are we missing? Oh, someone mentioned organelles, and this one, yeah, it is in there. This is where I say transport, because it's in probably,
Oh, like nutrients.
yeah, cellular transport. There are proteins, move stuff in the cell, and there are proteins move stuff in and out of cell. So there's intracellular transport and intercellular transport, and they do keep making a YouTube video taking it down. Let's see if I can find another copy. Because I'll put the notes back up in a second. Cell, transport, animation. Because they made up an none of those. Walking. Ah, there he is. That's my cute little guy. Absolutely beautiful. So this is a three-dimensional reproduction of how we think things are actually transported. Obviously, you can't see this, but this is based on calculations on how the proteins would let go and reapply. So that little guy, Fred, is hauling a huge vesicle.
My Fred.
All things I don't know the name of are Fred. Just like for a certain person who confused me for a long time, all seals are named Sparky. And we were at it, they lived in Comox, and they saw a seal. I'm like, oh, there's Sparky. And I thought they actually knew that seal, and its name was Sparky. It was only when they mentioned that at Victoria that I realized, no, they just call every seal Sparky. So that's Fred. Fred is a protein. By the way, this tube is also a protein. It's a transport hub. They form, and so it will self-assemble a set of it, make a long thing. Fred will walk along it, and then it will disassemble beside, and they're being made and broken up all the time. This is based on our best calculations on how they attach and let go. And the only thing that's really incorrect about this is how slow it's going. Because Fred would actually be like, and they'd be moving all over the time. They'd be super fast. This is a very slow reenacted, so we can see how it's moving. And it's literally just walking along.
Catwalk.
Yeah.
Fred's got that.
Fred's got a strut. Yeah. So motor protein, these are probably actin filaments that are guiding it where they're made and removed depending on where the cell wants stuff. So that's how things get moved around the cell. They don't have little motors that go there. They actually have Fred, who is a motor, who carries them there, or not carries, pulls them there, hauls them there. So there's a lot of the cell structure. And then on the outside of the cell, the cell membrane, there are gateways in and out of the cell. Those are individual proteins in the cell. So you need glucose in your cell. A protein has to grab it and take it in, or open up and allow only that molecule in. You don't want to let everything in. Otherwise, you'd just be everything. If everything could come in, there would be no difference between inside and outside. And then last one allows me to stop here and not mention all the different things because I could turn this into a whole biochem class and go through the 8 different functions or 9 different functions of proteins and all the cool things they can do. And I just say this last one is survival. And then there's a bunch of things that they have to be doing inside your body for you to survive. So the first one is blood clotting. That is a protein cascade because it is amazing that your blood isn't clotting right now. You should all be having multiple strokes and heart attacks. Because there is a system, a hair trigger system in your blood that's just ready to clot. You get cut, it clots right away. And yet it still doesn't clot inside of your blood. It is very tightly controlled, and all those proteins are just there, ready to go at a moment's notice. They come out, they get exposed to a variety of different factors from cut cells and a little bit of extra oxygen, and boom, you get a cascading effect, and essentially an instant Band-Aid appears on your skin. It's a really cool process that we do not have time to go into. In addition to that, it makes some hormones. So, I should check. What's a hormone? Not really much point telling you that it makes some of them when if you don't know what it. Have you not had that talk from like the health center? At a certain stage of your life, your body will change, and it's due to hormones. Everybody had some, yeah. So, actually not those hormones. Most hormones are actually made of fat. Estrogen, testosterone, the sex hormones are made of fat. They're a particular type of fat, same type of fat that cholesterol is based on. But there are some protein hormones. An example of that would be insulin. And a hormone is just a way of signaling your entire body by releasing a signal into the blood to do something. So your blood goes everywhere, so a very quick way to send a message is just to release it into a blood. A hormone is the chemical signal. So some hormones are made of proteins, which allows us, for the case of insulin, to make it fairly cheaply using yeast. We just put the gene in the yeast, you tell the yeast to make the insulin, it makes human insulin, and then we can inject that into ourselves if you have diabetes for a really low price, provided a billionaire doesn't get a hold of it and starts charging $200 per vial, because you can make it for less than $1 for the amount that you need for a day. That's probably it about hormones. Does anybody know what we did for insulin if you were diabetic before we could make human insulin?
Amputations.
Well, I mean, that's the effect. How did we get insulin? Because we knew that insulin was the treatment before we could make insulin.
We took
Dead bodies, if you were rich.
we took it
Dead human bodies, if you were rich.
from
Animal bodies, so you'd use like horse insulin from the carcasses of horses, if you weren't rich. And it was either way still very expensive. There's only so many horses that die, and so it was the breakthrough to make insulin, not heroin. Don't take heroin if you're diabetic or you know anybody who's diabetic. Was a huge
Oh
breakthrough.
my God.
Yeah. Next one, antibodies. You can make antibodies against a huge number of antigens. Part of virus, bacteria, antibodies attached to. I won't say too much about this either because, again, beyond the scope of the course. But you make antibodies, and so the first time you see a disease, you may have a cell, like one cell that recognizes it, and then after that, that cell just continues to make antibodies against it for the rest of your life. And so when you're invaded again, your body goes, I've seen this before, it's immediately attacked by antibodies, and for most of the time, you don't even get sick. This is the whole point behind vaccines. You essentially show the body, here's some antigens, and then they'll make antibodies against it. You just have to show the body in a way that doesn't make the person sick. So you do it with a vaccine, and then you can do that. We eliminated smallpox worldwide just based on that. A horrendously debilitating disease. If it didn't kill you, it definitely harmed you. And lots of people died due to smallpox epidemics that would occur periodically. And so we gave everybody in the world the vaccine, and it died out. We don't have it anymore. There was another.
What about monkeypox?
That's an entirely different pox.
Yeah, but
What
wasn't
do you
that like
know
a
about
thing?
monkeypox?
It's a pox.
What's your
It's a related virus.
Have you seen it?
I have not seen it. What's going on in the world news about monkeypox? Do we have a new pox?
It's
No,
in
it's-
China.
Yeah.
Oh. One of the main problems with
Yeah.
I will take an aside for a brief moment then to explain how some of this works and why why animals are such a danger. If you wanted to make a vaccine, an old method and one that still works really well is that you take the thing that you want to be protected against, say smallpox, and you grow it in an egg, like a literal chicken egg that's kept warm. You grow it in the egg. And it grows. Not well, because it's made to adapt to the human body, and the chicken egg does not have the same circular nutrients, but it's enough that it can grow. And then you take that and you put it in another egg. And then you take that and then you put it in another egg. And then you take that and you put it in another egg. By process of evolution, after 40 or 50 generations, what will that virus be really good at invading? Eggs. Or chickens. It would be terrible at invading you, and then they give that to you. And your body goes, this is a real easy enemy. Snap, snap. You're just good at invading eggs. Smack, smack, and then the virus dies, but you're still exposed to all of its antigens. You make antibodies against it. And then when the real smallpox shows up, your body just says, oh, we've defeated your less able sibling before. You're an idiot, and just kills it. The problem is that the reverse can happen. So you can have a virus that sits inside of another animal, and it's really good at invading that animal. But because they mutate all the time, one may mutate that's just kind of bad at invading, say, a monkey. But monkeys are close enough to us that once in a while it actually turns out to be better at invading humans, and then it jumps from one animal to humans. So they're evolving all the time. Vaccines is when we use the process of evolution and making them to make a vaccine, but the reverse can happen too. They can evolve just to get better in another species. And if we're around that species all the time, that just increases the risk that that can happen. So the swine flu coming from pigs, common, because humans raise pigs and we raise a lot of them and we're in constant contact with them. Just takes one farmer, someone working with pigs in it, and in general have them as pet, and it jumps to a human. It evolves a little bit, a few generations to get good at invading humans, and maybe it has a unique mechanism, spreads to the population. Bird flu is always one. We can't kill all the birds yet. Once we can, we'll be a lot safer from all these bird flus. But until then, we're just always in contact with birds because they poop on us from the sky, and so we just get their viruses that way. So yeah, we're always worried about jumping diseases from animals to us because it's kind of the reverse of a vaccine. They're testing themselves out in other animals, and then sometimes they'll just jump to us. This is why they're worried about. I think there were certain markets they're worried about just because they have a lot of bush meat. They'll have meat that people have hunted, and that's bringing new viruses into a marketplace in contact with humans. So that doesn't fill me with great joy. One of the vast improvements on that process is that takes time. Grow it in an egg, grow it in another egg, grow it in another egg. It literally years to get a good vaccine. COVID, we were so lucky because now we have sequencing. So researchers in China who were first exposed and knew about it, sequenced the virus, spread that to every lab in the world, like here's all of our data. Every lab in the world immediately started working on mRNA viruses. And I'm explaining this now because we're going to be doing mRNA very shortly. It's on the next slide. And that allowed people to start not growing it in eggs, by taking out parts that coded for antigens and synthesizing a vaccine from scratch. That's a much faster process if you can do that. That lowered our vaccine waiting time from years to months. It was such a huge improvement. If COVID had happened 2 years earlier, we wouldn't have had the tech to do it. We were just extremely lucky. We would have had the full 1918 whatever plague equivalent going around the world. As it was, we had a fairly bad experience in terms of how many people died, but a lot fewer people died because we got the vaccines out so fast. Total props to the researchers who did that. They didn't have to. Science is all about sharing your research, and because they did, it helped everybody out worldwide. So lucky. Some of them even got in a little bit of trouble because companies are like, well, we could have charged more for it if we were the only ones that had this. But as it was now, we had multiple competing companies with different vaccines. We had a chance to pick the best ones. So we're doing protein synthesis. That's the way we create protein. I should have asked you what synthesis meant first, but now I've defined it. It takes place in 2 stages: transcription and translation. And those words are picked not arbitrarily, but based on their normal English meaning. So, get to block B here. I'm going to ask someone what does it mean to transcribe something? See who? Point. Transcribe.
Move something.
Move? Either transport, you might be thinking of.
Transfer.
Transcribe. It comes from the word scribe. Don't know if you've encountered that. We're not scribble. Wrote that wrong.
Isn't it?
Another way to approach it might be, what does a scribe do?
Right.
Right. They copy. Earliest scribes, all they did was copy, whatever work that the people put in front of them, and just copy this because we don't have printers and we need to make copies. And so scribes just copy things. So to transcribe is the process of just copying. So transcription is copy DNA to RNA.
You might want to write it here.
And, sorry?
Like,
Oh, I'm going to.
okay.
This is sort of the rough introduction, then we do it in more detail. In fact, knowing that, I'm going to leave that off. And then translate. What do we mean when we say translate, Jaden?
Well, I'm assuming like
Perfect. These are much the same. Translate is RNA to protein. And this, they're different, very different. And these are the same. What does a squiggly line like that mean in math? Approximately. So this is like approximately, like really, kinda. This is like approximately equal to, one line is like even more error, approximately. So DNA to RNA, they're both kind of the same thing, DNA and RNA, whereas protein is vastly different. It is not the same kind of molecule as RNA, and it will look and act completely different. It's made out of different stuff, whereas these are kind of made of the same stuff. As we talk about this topic, I do have to mention, I used to have a good diagram. I learned this morning, I think the diagram's gone. Amanita biosporigia. There's a related species on Vancouver Island. It is incredibly rare. Apparently someone once found of these exact ones on Vancouver Island too, though. Stupid people. Where's the map of its worldwide distribution? It's mostly eastern side of North America. However, let's go down to where you don't need to know what it looks like. Similar species, taxonomy, toxicity. There we go. The toxin inhibits RNA polymerase II, thereby interfering with DNA transcription. So you can no longer transcribe the message from DNA to RNA, which means you can make no more protein. So once you've eaten this, you are making no more protein for the rest of your now very short life. Because you can't even repair the things that are broken because you can't make the machines, the enzymes that would repair them. So you're mostly going on momentum now with all your road proteins. And about I think it starts around 4 to 6 hours afterwards, every cell in your body starts to die because there's nothing keeping anything going anymore. You're not making anything new. And so that's how long the old mRNAs keep working. And then the treatment is to put you in a coma. That doesn't extend your life at all. That just helps the people in the room because your screaming is incredibly distracting to all the other patients because all of your cells start to die. So every nerve in your body starts telling you pain because it wants to tell you that the cells next to it are dying, but it's every cell in your body. So it's an unbelievable amount of pain. Heroin and our strongest painkillers don't touch it. You have to be put into a coma so you don't scream until you die.
So you're cooked.
You're totally cooked. And speaking of cooked, we actually know it has a very pleasant nutty taste because it takes 4 to 6 hours to start, so you can talk to the people beforehand. And they'll say, oh yeah, I cooked it. And they'll show you the pan and you'll see the mushrooms in there. And I'm like, how did it taste? Oh, it's really good. Is this bad? And you're like, yeah, it's pretty bad. Don't eat mushrooms unless you know where they came from. It's not like, there's plants that are poisonous. But mushrooms just seem to have this whole like, I'm perfectly fine, you can eat me and I will kill you and all of your children attitude. Whereas at least with plants, you'll realize it and sometimes and throw up first. Not all plants. Don't believe everything I say because I'm not but I would be extremely skeptical of someone who's foraging for mushrooms unless I know them and know they've been trained or it's gone through some sort of growing process where they know exactly what they have. Mushrooms are just wild.
Does the dosage of that toxin matter?
Pardon?
Does the dosage of that toxin matter?
It's an extremely small amount.
Yeah.
I think enough from one mushroom can kill you. Where's what opened up? Not that one. Was it? Did I close it? I closed it. When we have another moment in the class, I will look that up because that's good to know. Is there a safe dose? I'm pretty sure, but I'd be curious. Like if I licked it, would I die? If I go around licking things, I'm probably going to die anyway. But that's not the point. 1. Where is your DNA located? Guy.
In the nucleus.
In the nucleus. So 1. This happens in the nucleus transcription. 2. We already said this on the previous stage, but copies portion of DNA to RNA. And this is a nucleic acid, and this is a nucleic acid. So they're the same kind of thing. That's why it's transcription and not translation. DNA is deoxyribonucleic acid. Based on that, what do you think RNA is? Ribonucleic acid. It's not a deoxyribo, it's just a ribo. That's the main difference. It's a different type of sugar. RNA is also not double-stranded. It doesn't have a double helix. There are other differences as well. But in terms of the base structure, it's a ribo, not a deoxyribo. And then, if it happens in the nucleus, where does that RNA need to go? Katie, where are proteins made?
In ribosomes.
In ribosomes. Oh, I should do a different color. Transport to ribosomes in cytoplasm. I'm not going to retest you in all the organelles, but the ones we mentioned in terms of where things are happening, you should kind of know. So you have your nucleus, makes a thing, your rough endoplasmic reticulum, you've got ribosomes on top of it, that's why it looks rough. Your smooth button, by the way, is what makes the fats. So your estrogen and testosterone will be made there. There's the Golgi apparatus, which packages it into things that you want. The cell membrane has the package of it, etc., etc. So ribosomes are primarily used to make that protein. Yes. What's that reminder? What time is it? 12:33. We started at 12-ish. 5-minute break. Go get a glass of water. Walk around. And we'll start about 12:30. Let's say 39. That gives that extra 30 seconds. And then everybody. No, you don't have to leave. Or you could stay and we just discuss quantum. Yeah, it's fine. Get a drink of water. One thing I don't like since COVID is that there's no more drinking fountains. If you forgot a cup, you're toast. Right? Excellent. Glad you agree with me. I take your silence as continued and undying agreement with everything. The world is flat. The sun is only a little farther away from us than the moon. And light speed is about the speed of sound. Excellent. Well done. You can tell that the moon is about the same distance from us as the sun because when they overlap in an eclipse, they're exactly the same size. That can't be a coincidence. Right? The sun and the moon are approximately the same size.
No.
When you have an eclipse, the moon is exactly.
Yeah, but distance.
Yeah, I know that's what people say. Are you telling me it's a pure coincidence that they just happen to be at exactly the right distances to make them look like the same size? Because that'd be pretty weird.
Gravity?
Gravity wouldn't change that.
No. If the mass was different in like the moon and the sun, then the gravity on each other would change and their orbit and position.
Their gravity isn't very much. Earth's gravity is bigger than both. That's why the sun goes around the Earth. Right, Jaden? Excellent, I'm glad you agree. Pardon? No, the sun is about the size of the moon.
Yeah.
Tiny.
Wait. So I'm nearsighted.
So you're saying
Isn't it just,
this
it could be
is
a
bigger
pure
than the
coincidence.
No. The moon is sizable. It's roughly a quarter of the size of the Earth. And so is the sun.
Is the distance of, it could be purely coincidence because that's how the universe came to be anyway too.
Sorry, what?
Well, if you're saying, oh, it's weird that it would just be a coincidence, but
Yeah,
Isn't a lot of stuff.
it would be almost unbelievable to think that unless you just believed everything you were told. But if you looked at the evidence of your senses,
You won't wait.
they're both
Yes.
about the same size, they have similar effects, probably about the same distance away.
like 15 minutes?
Saying that one is unimaginably farther away and gigantic and huge
Just me and Ace.
seems weird. Seems weird just to make that up.
Yeah, but entropy.
what?
Entropy is the increasing randomness over time. How does that?
Yeah, because it's random.
Pretty cool.
Quantum. One of the fundamental things that makes quantum interesting.
Yes.
You've heard that both light is both a particle and a wave. And we know it's a particle because if you shoot it at stuff, it will break things off like a particle. So it is a particle. But if you take things that are particles,
So I like left it.
and let's say you literally have a gun and you shoot bullets at a couple of holes, you'll get splattered patterns based on how they come out of those holes. Light doesn't do that.
Why do I look like someone who would have to?
Did you ever do like wave tanks in grade 7 or 8 and watch waves?
Oh, yeah, like the wave.
Yeah, if a wave goes through a hole, what happens to the shape of the wave? If it approaches it flat, what does it do if it comes through a narrow hole?
Goes on.
Do you have any food?
It'll like rings out from the hole as it spreads out.
Oh, I did not know.
Oh, it doesn't. If you have two holes,
That's cool. Are you playing tomorrow? You playing tomorrow?
Yeah.
there will be areas where you have one wave
It's a senior year.
and another wave overlap. Then you get a large wave.
Oh, it's
Great. If you shine light through this, you get exactly the same thing. Because where the wavelengths of light overlap, it's really bright. And then where they don't, where they sort of have a trough and a wave, that equals that cancels out, and so you'll get a dark area. So you get dark, light, dark, where things overlap and don't overlap. So it's a wave.
Oh.
Except if you have those two holes and you rig the experiment up so you can see it going through, like you can detect it, then you don't get that, then you get the splatter pattern. So it only acts as a wave if you're not looking at it. Yeah, that's weird. And
That's very weird.
in that process, we only shot one photon of light. So it had to choose which hole it went through. But even though there's only one photon, it can't interfere with itself. You still see the same pattern. It does interfere with itself. But if you look at it to see which one it goes through, it only goes through one of them, and you don't get that pattern. That's why quantum is so weird. I think I went past the time. No, it's exactly on time. That's perfect. Well done.
Yes.
Yay. Okay, translation. I ended up taking up too much space for this, so I'm going to go like that and cut it off. And for translation, happens. I should make an @ sign. This is notes. At ribosomes, as we've said. The mRNA is transformed and a peptide a peptide, a single strand of amino acids, a small protein. And I define that because you'll see the word peptide come up, and all they really mean is small protein. The reason why there's a different word is the proteins that are eventually packaged and used by your body. Some of them are peptides. Some of them are actually multiple proteins connected together to make a larger protein. And so some proteins are made and folded and done, things are packaged in a way so that it has more than one protein attached to it. And so that entire process is a bit beyond. But you'll see peptide, and what they mean is a protein coded for by one gene. It's a continuous strand of protein. So a small protein.
What does that say happens OT ribosomes?
Happens at.
Oh.
It just says uppens. What was that? Ah, ha, yeah. Damn apples. It read it. Oh, beautiful. Look at that. The mRNA is transformed.
What's the difference between RNA and mRNA?
Ah, I'm glad you asked. What does the M stand for?
I have no idea.
This type of RNA is called, we'll go right to the bottom, mRNA, which is equal to messenger RNA. So we're going to see mRNA, tRNA, T stands for transport, and rRNA. R stands for ribosome, so you need a little bit of RNA in there. And it's created by complementary base pairing with DNA. Okay. Good. Can I scroll back up? So to explain what's going on here, people just objected to my handwriting. I will try something new. Well, first I'll write this. So first thing we're going to do, transcription. And let's cut this part off because that goes with the translation from before.
Is that you a typo or is
Where?
it says pink ace?
Here?
A-C-A-U.
No, it's not a typo. Vicious lies. Let's go there. What color should we make it? Orange. DNA is unwound with the enzyme. What's the name of the enzyme that unwinds DNA? That unwinds the helix. Histones are what DNA wraps around. What do enzymes end with most of the time? Ase. So there's lactase. Anybody have an unwind
Protease?
helix? Pardon?
Proteases?
Protease digests proteins.
Oh, helicase.
Helicase. Nicely done. Oh. And I should put a number 1 in front of that. So the DNA is unwound with enzyme helicase. We can't see that very well, so let's go down a little bit. and then 2 complementary base pairing of mRNA nucleotides. 3 nucleotides join and make mRNA. Now there is an error on here. That I only discovered last class. This and that should be reversed. This is 5 prime, that's 3 prime, and that's 5 prime, and that's 3 prime. And my main reason for doing that, if it goes 3 prime to 5 prime, it goes 5 prime to 3 prime, and that makes 5, and that makes 3, and I'm not necessarily going to expect you to memorize that, but DNA is always read in the 5 prime to 3 prime direction, and we're reading this one. This one at the bottom is the reading strand. And you always read from the 5 prime to the 3 prime direction. Those primes, the little dash on top, refer to the direction the sugar is pointing. So one of the extensions last day in the lab was that you could show that the sugars face different ways. So the strands literally point different ways. There is a reading direction. The same way I, for example, read from left to right. DNA is always read from 5 prime to 3 prime because you can rotate DNA all the way around. It's a 3-dimensional structure, so which way do we read it? Well, you'd read it this way. If you're reading this strand, you would read it that way. So first off,
Is it always 5 and 3?
you always read it 5 to 3.
Like no, but like why those numbers?
The 5 prime and 3 prime has to do where what end of the sugar is hanging out of that strand in terms of like the phosphate and this. So are you looking at the 5 prime or 3 prime type of thing? Because it'll face different ways depending on which way the DNA is facing. So it refers to the structure of the sugar. We don't really have to worry about it. But mostly so that when you get to university, if I actually taught you the wrong way around, it would be embarrassing, and you'd be like, my Science 10 teacher said it was the other way. My diagram was wrong. So we first have, let's draw, oh, wrong color than I selected. Helicase comes in, unwinds it, and opens it up. We're now making this. You probably actually can't read that. That's supposed to be a T. So T goes to A. Complementary based pairing. T goes to A. G goes to C. And then T goes to A. And A goes to U. RNA does not have thymine. It has uracil instead of. So RNA, U, not T. So it is made of nucleotides. It has a different nucleotide. It doesn't have thymine. It has uracil, and so that is one of the difference. It also makes a single strand. And this would also change because we're making it in the 3 prime, because that's 5 prime to 5 prime direction. And it will continue doing that until it's done, and that will be released as it is. There is an enzyme involved here. I'm not going to name it. You can learn it later. But it's RNA polymerase, just making a polymer of RNA. So we put them in one at a time. RNA polymerase will come in and join them all up, and then it will be removed. So to label it, this is 1, and then the pairing is 2, and then the structure being joined together is 3. Differences between DNA and RNA. Already RNA has which one? Uracil. So uracil just replaces thymine. What other differences about it compared to the DNA in terms of what it looks like?
The sugar is different.
Ribose in RNA.
Single strand.
You did one, so you did the other, single strand. That's all the differences in, DNA just has one type. There are 3 types of RNA. There is the one we've just discussed, mRNA. There is tRNA, and there will be rRNA. Also, RNA in cytoplasm and nucleus, DNA only in nucleus. Okay. Now, couple of restoring it. Good. Everybody still ready? Okay, next page. Next page, you do it. So at the top, I've got the DNA. Write the equivalent mRNA below it. Then we have, for this DNA, write the equivalent on this. And here, this will be our reading strand. Then use that strand to make that strand. Oh, there it is. So 5' refers to that, 3' refers to that, and which one it's attached to the phosphate group by. So when you're leaving off a nucleotide in one direction, it's going to have that sticking out. If it's at the bottom, it's going to have that sticking out. Ooh,
Wait, so like for the bottom one, we don't write anything on double strand, right? We just write the single strand of mRNA. Like
first, you put in.
we match the. Okay.
First, you put the complementary DNA nucleotides here,
And then we.
and then you would use this as the reading strand to make the complementary nucleotides for the mRNA.
Okay.
For the top, with the pink colors,
Mhm.
do we put what the codes are in DNA?
Nope, for these ones, I want the mRNA.
Yeah.
Put
Basically,
what there are in
right.
mRNA.
For the mRNA, yeah.
A is still A,
Yeah.
but T
Oh, yeah.
is U.
Right.
Okay.
Yep.
So that's for.
So if I did the first two, A goes to,
A is U.
yeah,
You're so.
T goes to
You. A.
A. T is U, but it attaches to A. G goes to and so on and so forth. Bottom one too.
Oh.
Yeah, yeah.
So let's say this is T.
Mhm.
This is A. So down here would be U.
T goes to U.
A.
Yes. Sorry, T goes to A.
And then A goes to
A goes to okay. Yeah. Even I messed myself up. T goes to A. Yeah. You, yeah. You got it first. T is
an animal here.
but T
Sure, it's a
belongs
nose. Those are two eyes. Yeah, that's what your head.
Oh,
But the
right.
nose is
The
ugly as
Yeah.
a. You see that?
So
Yeah.
your reading frame is the other frame.
Why are you backing up?
Well, why is
Yeah, why are you doing that?
I'm putting away my laptop.
And your notes are closed.
Genetic failure. Genetic mutation. Genetic mutation.
Yes, sir.
What's that weird picture?
Oh, I told AI to give me a picture of cute mutations.
Oh God.
Mm.
Yes, dude. Yo.
Mhm.
Wait,
So,
Wow.
search up another one.
That's crazy.
Pardon?
It is
Search up
crazy.
another one right now.
search up. No, I like told AI. Like
Talented. I know.
it made it for
That
me.
should
It's
be
bespoke.
extra. That is
Can you
fantastic.
search up cute mutations?
Cute mutations. I'm good.
Right now.
Ooh.
Can you do it right now?
Will it?
Can you go to images?
Be like, yo, that is
I
not.
like,
I like that
this
one.
Oh.
That's the. That's from propercooking.info. Yeah, that's real cute there. Oh, there, cute. Lobster or like 3 eyes.
Yo, that cat has a second face.
I like the first one.
Oh, that. Oh.
Have you seen the dog with a tail coming out of its forehead?
No, is that a real one or is that
Yeah,
AI?
yeah. Look up Narwhal the dog or something. I think.
What?
I think it's Narwhal the dog.
Oh, so cute.
Jesus. That does not suit.
It's kind of nasty.
Wait, does it wag?
You're leaving in 2 seconds, so I shouldn't be too cute. Just to double check your work, I think I saw everybody. A goes to T, and then T goes to A. T goes to A, and then A goes to U. When you've done this strain, you're reading this one, but which one should it look the most like? Like, which one should it be a clone of?
Top one.
The top one. A, every T, so you can always check it against the other strand. You're good to go. Thank you. There we go. And I will, stop the recording.